Namaste all. I, Javian Shalini Devi Prajapati from Faculty of Education and Methodology Department of Food and Biotechnology. Giving this video lecture on 19th September, the on the topic of overview of the immune systems and cell and tissues involved in immune system. So firstly we started our topic overview of the immune system. So what is immune system and what is immunity, as we studied immune system usually in order to defend multicellular creatures against infection. We call immune system, or we can say our immune system developed to defend against the multicellular creatures against the infections. Our immune system protects the body from a variety of invaders, including use parasitic, kidney worms, and blah blah blah, which length in over 100 centimeter to 100 nm, or a microorganism and pathogens and cellular viruses that cause polio or other viruses. This is our immune system, but if we talk about immunity, so immunity refers to a condition like resistance to infectious disease. Comes from the Latin phrase immunes, which means exempt. In 15th century, some Chinese and Turkish were performed induced immunity. This is our first recorded attempts in immunity or immune system, we can say. Next, after attempting to prevent smallpox is a kind of disease which percentage from 30 percentage cases that leaves the survival disfigures for life in ancient approximately 15th century. Smallpox is a kind of disease which recorded by Edward Jenner. Uh, Later, with a railway to focus on smallpox, the English physician Edward Jenner achieved a significant advancement on the place plant developed to immunity. Means our immunity will develop by or like vaccination process developed by Edward Jenner. He developed a smallpox vaccine from using cowpox vectors spread across Europe. Contribution of much more sciences in ancient like Edward Jenner, Louis Pasteur, and many other blah blah blah. Edward Jenner discovered about the smallpox vaccine, and our Louis Pasteur established the immunity of cholera by injecting the bacteria that cause for cholera into hands that later required deadly cholera actually. So Louis Pasteur was able to establish that he had successfully growing the bacterium in culture media. And after he and his colleagues resumed their study after their summer break by injecting several hens with an old bacterial culture. Then hen were sick, but Pasteur was surprised to see them get better. So Pasteur developed a fresh culture of the bacteria after becoming integrated with the idea of injecting some fresh untouched hands with this deadly concoction. Then, however, as the tale goes, he had a limited number of fresh chickens, so he utilized combinations. There's some immunological research, like some scientist or research on the basis of in 1901 Emil von Behring from Germany, he researched about serum antitoxins. Next, after 1905 approx, our scientist Robert Koch from Germany developed the cellular immunity to against to tuberculosis. Tuberculosis refers to TB means tuberculosis bacteria which cause TB. Next, after duration between 1908 approx, Paul Ehrlich from Germany, he developed the role of phagocytosis and antitoxins in immunity. Uh, in 1913, Charles Richard from France developed the anaphylaxis process, which comes under immunology. Next, after in 1919, Julius Burger from Belgium developed the complement-mediated bacteriolysis. 1980s, your snails, chicken, dustrets, birds, scientists from United States, France, and United States, respectively, developed the major histocompatibility complex (MSC complex), which also comes under our immune system. This way, immunology grew. Like in 1991, E. O'Neill Thomas and Joseph Murray from United States, the board researchers from United States, they developed about transplantation immunology, and some of the scientists in 2008 like Zorho Sand and Francisco Barry Sanchez developed the role of HPV in causing cervical cancers and discovery of HIV. All about our history regarding to immunology or immune system, how to start about immunity and immune system. Then after we studies about what is actually immune system and cells and tissues and organs involved in immune system. So next topic over on the behalf of cell involves in immune system. Formation of immune cells or of human blood cells is comes under hematopoietic process, and there are some stem cells which formed into in bone marrow. And actually we can say formation of blood cells start from bone marrow, and these are divided in two types of progenitor. First is common myeloid progenitor and second is common lymphoid progenitor. Myeloid progenitor refers to in common myeloid progenitor comes under some erythroblast, potato cells, precursor, myoblast, dendritic cells, monoblast, and megakaryoblast. So, whichever erythroblast, erythrocytes. Erythrocytes refers to RBCs, which are parts of our blood cells. Next, after megakaryoblast, form megakaryocytes, and then after it's converted to platelets including some blood clotting factors which comes under blood cells. Next, after our potato mast cell precursor which formed mast cells. Mast cells is a kind of some specialized tissues or cells similar to basophils that can trigger some local inflammatory reactions in our body, and also responsible for some allergic reactions. Next, after our myoblast cells is also parts of common myeloid progenitor. So myoblast cells divided into further three types of cells, which are erythrocytes, which are eosinophils, basophils, and neutrophils. Eosinophils, basophils, and neutrophils we also studied in these all cells comes under WBC's parts of granulocytes. So functions of eosinophils we can say they are active in worm and fungal infections. Means if any kinds of fungal infection and worm infections occurs in our body, then eosinophils from blood cells, they work on that infection and also assist in some allergic and inflammatory reactions. Then after we we studies about basophils. What is basophils? Is also a parts of common myeloid progenitor which are formed after during hematopoietic process. Basophils also acts in inflammatory events and allergic reactions. Then after our the neutrophils, what are neutrophils? Neutrophils functions is in essential blood phagocytosis. Also active and engulfs and kills the bacteria and perform a phagocytosis process. Next after dendritic cells and next after monoblast. Monoblast is also part of common myeloid progenitor. So monoblast divided in further monocytes, next macrophages and dendritic cells. Monocytes are the kinds of blood cells which blood phagocytes that rapidly leave the circulation and they further mature in macrophages and dendritic cells. So next after macrophages, these are largest phagocytes that ingest and kill foreign cells, strategic participants in certain specific immune response. Next after we study about dendritic cells that are also parts of common myeloid progenitor. These resides throughout the tissue responsible for processing foreign matter and presenting it to lymphocytes. This all about common myeloid progenitor. Next, after we can say about common lymphoid progenitor. So, common lymphoid progenitor are of three types mostly that are there are lymphoblast, NK cells. NK cells refers to natural killer cells and innate lymphoid cell precursor. Lymphoblast further perform lymphocytes and lymphocytes further classify into T cells and B cells. Which are our lymphocytes? They are primary primary cells which involves in specific immune response against foreign particles, pathogens and microorganisms and invaders. What we say. Lymphocytes divided into two cells, T cells and B cells. T cells perform a number of specific cellular immune response such as assisting B cells and killing foreign cells or foreign particles, pathogens like microorganisms, blah blah. Next, B cells, which are all B cells, differentiate into plasma cells from plasma cells and form antibodies that are parts of humoral immune cell response, and which are T cells is a part of cell mediated immunity. Next, we discussed about natural killer cells or NK cells, which are our natural killer cells. They act on kill tumor cells and cell with intracellular infections. Next, after we studied about innate lymphoid cells. Innate lymphoid cells is a specific kinds of cells which secrete the cytokines that assist to both in innate immunity and adaptive immunity. Innate lymphoid cells are not antigen specific, support lymphoid tissue development. So this is all about our blood cells involved in immune system. Next, after we discussed about organs involved in immune system. So organs are divided in two types. First is primary lymphoid organs and second is secondary lymphoid organs. Primary lymphoid organs in comes like thymus and bone marrow, and which are our secondary lymphoid organs. There are all lymphatic system regarding to spleen, blood vessel, lymphatic system like spleen, lymph node, and lymph vessels. This all which are our primary lymphoid organs. Primary lymphoid organs we discussed yesterday here about thymus glands. In thymus glands, maturations of T cells occurs, and which are our Actually, we can say T cells maturations occurs in thymus, and formation of T cells from bone marrow. Next, after which are our B cells. B cells maturations occur in bone marrow. Also, they form from bone marrow. This is all about our short lecture on immune system, overview of immune system, in which we study the body's brain and cells involved in immune system and organ and tissue involved in immune system. Thank you.